Good morning, welcome to my channel, or welcome back if you've been here before. My name is Beth Ann. Normally I am a booktube only, books all the time channel, but as some of you who've been around on my channel for a while might know, I am a bee scientist. I am a full-time research scientist at the University of Minnesota. And this week I came back to campus. Normally I live in New York and I work full-time from home these days, but I'm here this week dealing with some of my bee specimens from the past 2 summers, getting them finally processed and curated before we collect new specimens for this upcoming summer. So I may not have time to make a booktube video this week because there's a lot to do, but I thought I would just do a quick sneak peek into one of the tasks that I spend a lot of time on as a bee scientist. So what I want to talk about today is the bees that are in this box, and we're just going to talk about one step in the process of getting bees from nature into a published scientific paper. So these bees were collected in the past 2 summers, and they were collected in a particular type of trap called an emergence trap. And what an emergence trap does is when we're trying to figure out where bees might be nesting, and actually here, I'll put up a photo of an emergence trap. When we want to know where bees are nesting, we can put out these little tents, and we can move them around throughout the summer to try to catch bees in different locations. And the idea is that if we go out in the evening, we place this tent, we are going to collect any bees that were actively nesting in the area under that tent and had returned to that site for the night. And we're also going to collect any bees that are emerging, are being born from a nest that was made either earlier that same summer or in the previous year. And that gives us information about what resources, what types of places our native bees are nesting. Now this is focused just on bees that nest in the ground or very near the ground, because these tents are near the ground. But in North America, that is the vast majority of our bees are what we sort of call ground nesting bees, which is a bit of a misnomer because they do nest in the ground, but they also might nest in the leaf litter and in the thatch of dead grasses, places like that. But we're talking about bees that are near the ground. So once they are in the trap, we can collect them. There's a little jar at the top of the trap. So we pull them out. They're all gunky from the fluid that they were in the jar. We wash them off and then we pin them. And in the first stage of pinning, we're just giving them sort of these messy labels. They have quick notes that were from in the field about where and when each bee specimen was collected. Bees that were collected in the same trap will just be in a row next to the same label. Each bee gets its own unique number as we're quickly going through and pinning these bees. Then the next step is, once we've got them all pinned, we will enter all of this information into a spreadsheet, which is the start of our database for the project. Once that spreadsheet is complete, we will use a special program that will assign barcodes to each bee. So in that spreadsheet, each row is a bee specimen, and they all get a unique identification and a unique barcode. And then we print out these permanent labels, which you can hopefully sort of tell look a lot more professional. And so at this point, all the bees are still mixed in together. So here are the bees with their final labels. And you can see we've got a mix of different types of bees. We have some larger bees, but even these are not as large as the bees you folks watching this video might be familiar with. You're probably familiar with bumblebees, carpenter bees. Those are all big bees, maybe even honeybees. And so some of these bees, like this one, and this one, are maybe the size of a small honeybee, but all of these little specks are also native wild bees from Minnesota in this case. And they're very hard to identify down to species. So even though I have a PhD in bee science, I can't do that because my PhD is in studying about the habitat and the plant associations of bees. So at this point, once they have their permanent labels, they go to a taxonomist. And we're very lucky here at the University of Minnesota. We have a bee taxonomist in-house. He works in our research lab. And a bee taxonomist is someone who he can identify all of these bees to species, but his job is actually more than that, and he's devoted his career and his education to becoming a taxonomist. It's a very specialized scientific area, and it's the study of how species are related to each other. And yeah, it's stuff I don't know how to do. But so he takes these and then he starts to sort them out. So he'll put all of the bees that are the same species will end up in the same little box. and then he'll assign species identifications to each specimen. So then we can go back through and we can scan the barcodes, which is a much quicker, more efficient way to get those species identifications entered back into the database. We can just scan the barcodes for all the bees that are the same species and just copy paste that species name into the database, scan the next batch, etc. And then once we've done that, these bees will get put into an insect collection. So like we have an insect collection, a small museum essentially here at the University of Minnesota. And then I will get the data and I will analyze it and write a paper with it. So this is the stage that I'm on in all of that is I've printed out these formal permanent barcode labels, and I'm just replacing the messy, sort of processing labels with these formal barcode labels. So it's a tedious, time-consuming task, but it's nice to be able to do it, for a week as a break from some of the more difficult work that I do. And I get to, catch up on BookTube a little bit and listen to some audiobooks while I do it. So I hope this was interesting, just a quick little snippet into a little bit of what a bee scientist does for a day. Feel free to ask questions down into the comments. I was just focused on just showing you one little piece of methods. Obviously, I didn't really talk much about the science of what I do, the research questions and things like that. Maybe we can do a later video. So anyway, just thought I would share. Thank you for stopping by, and I'll see you all in the next video. Bye.